Hello viewers, the topic of today's class is enzymes in biological systems. The objectives are to understand the importance of enzymes in biological reactions and to acquire knowledge about the functioning of enzymes. For convenience, the content has been divided into 5 modules. To go on to the first module, enzymes can be defined as catalysts of biological systems, which are remarkable molecular devices that determine the patterns of chemical transformations. They also mediate the transformation of one form of energy into another. To go into the different types of enzymes that are existing in nature, the first type are a group of enzymes called the metabolic enzymes. These enzymes are primarily in charge of energy production in the body. They also help to detoxify the body on a cellular level and even help our sensory system respond appropriately. The second type of enzymes are the digestive enzymes. These enzymes include assisting the body break down and assimilate the food that we eat into nutrients. The third type of enzymes are called the food enzymes, which primarily come from plant sources of food. Our body does not make these enzymes, but they are present only in the food that we eat. Enzymes are destroyed by heat, therefore, when we cook food, they are partially denatured. It is therefore important to include some amount of raw, fresh fruits and vegetables into our daily diet to get the benefit of these food enzymes. Next, we go on to classification of enzymes. Many enzymes have common names that provide very little information about the type of reactions that they catalyze. For example, a proteolytic enzyme secreted by the pancreas is called trypsin, which does not give any indication about the type of reaction it catalyzes. Most other enzymes are named for the substrates upon which they act and for the reactions that they catalyze, and they end with the suffix ASE or ase. Thus, an ATPase is an enzyme that breaks down ATP, whereas ATP synthase is an enzyme that synthesizes ATP. To bring some consistency into the classification, in 1964, the International Union of Biochemistry established an enzyme commission to develop a nomenclature for enzymes, which is common. Reactions were divided into 6 major groups numbered 1 through 6. If you look at the table 1, you can see that oxidoreductases catalyze oxidation reduction reactions. For example, lactate dehydrogenase. Transferases are involved in group transfer. Hydrolases catalyze hydrolytic or hydrolysis reactions, which involves transfer of functional groups to water. Lyases add or remove groups to form double bonds. Isomerases isomerize intermolecular group transfer, while ligases ligation or formation of 2 substances at the expense of hydrolysis of ATP, which means joining of 2 substances. Now, enzyme classification is based on either of these classes. So, each enzyme is assigned a 4-part classification number and a systemic name, which identifies the reaction it catalyzes, which is called the enzyme commission number E.C. The first number denotes the name of the class. Each class from 1 to 6 described in the table is again divided into subclasses based on the reactions catalyzed. The second number indicates the subclass, that is the type of group in the donor compound which undergoes oxidation. For example, denoting a CH-OH group, 2 denoting an aldehyde or ketone group, etc. For transferases, it indicates the nature of the group that is being transferred. The third number indicates the type of acceptor group that is being accepted by a compound. For example, a phosphoryl group or a hydroxyl group. The fourth number denotes the compound that is accepting the functional group. So, a complete list and description of thousands of known enzymes is maintained by the Nomenclature Committee of the International Union of Biochemistry and Molecular Biology. Enzymes are highly specific both in the reactions that they catalyze as well as in their choice of reactants, which are called substrates. An enzyme usually catalyzes a single chemical reaction or a set of very closely related reactions. The specific site on the enzyme where a substrate binds and catalyzes is called the active site, and each active site has a shape that fits a specific substrate or substrates only. Since the enzyme is a protein and since proteins contain amino acids, side chains that attract the enzyme's particular substrate are functional groups of amino acids which are projecting out into the active site. So, side chains are specifically positioned to speed up the reaction and specifically bind to the substrate. For example, chymotrypsin's enzymatic function is to accelerate the breaking of the peptide bonds that link an amino acid which has a non-polar side chain, for example, phenylalanine, to another amino acid on the interior of the polypeptide chain. Look at this figure, you can see the active site, you can see the catalytic site, and you can see the specificity for binding to a non-polar group on the amino acid chain. So, scissors indicates where the enzyme cuts the peptide bond. We have listed out all the three enzymes in this figure, like trypsin being specific for lysine or arginine, chymotrypsin being specific for a non-polar group, and elastase being specific for a peptide bond between alanine and glycine. Now, if you look at figure 6, it shows the similarity between trypsin and chymotrypsin when superimposed on each other, indicating that structural similarity is just not enough for the commonality in substrate binding. The enzyme substrate complex called the ES was first proposed by Charles Adolphe Wurtz in 1880, and it is central to the action and specificity of enzymes. An important principle here for specificity is the weak binding interactions between the enzyme and the substrate, which provide a substantial driving force by adding energy to the reaction, and this binding force helps in breaking the enzyme substrate complex and converting it into enzyme plus product. So, the binding energy accounts for a huge rate accelerations which are brought about by enzymes. The energy available from formation of a single weak interaction is generally estimated to be about 4 to 30 kilojoules per molecule. This particular figure shows you that the enzyme has to climb a hill which is called the activating energy, and this energy required by an enzyme to climb the hill is reduced substantially by the binding energy. So, the overall energy available from a number of such weak interactions is sufficient to lower this activating energy by 60 to 100 kilojoules per molecule, which are required to explain the large rate enhancements observed for many enzymes. Now, look at this picture. It shows you the enzyme active site and the substrate. It shows that the active site conformation is very different from the substrate conformation. But once the substrate moves closer to the enzyme has a capacity to conform to the substrate and change its structure and conformation to that of the substrate. This is called the induced fit model of enzyme activity and is very inherent to specificity of enzymes. Now, we move on to module 3 and we will deal with coenzymes or cofactors and isozymes. The catalytic activity of many enzymes depends on the presence of small molecules called cofactors or coenzymes. Such an enzyme without its cofactor or coenzyme is referred to an apoenzyme. The whole complex of enzyme plus coenzyme or cofactor is called a holoenzyme, and the protein portion or the enzyme portion alone is called the apoenzyme or the apoprotein. Look at the picture which denotes the presence of the catalytic site into which the substrate has bound, and you have the coenzyme and cofactor closely associated with the catalytic site. Cofactors can be subdivided into two groups, metals and small organic molecules. Essentially, inorganic ions that aid in enzyme catalysis are called cofactors, and the small organic molecules which are mostly from the class of B complex vitamins are called coenzymes. Enzymes that use the same coenzymes are usually mechanistically similar. Examples of common coenzymes are ATP or adenosine triphosphate, or the currency of energy used in the body, NAD nicotinamide adenine dinucleotide, FAD flavin adenine dinucleotide, FMN flavin mononucleotide, pyridoxal phosphate, biotin, thymine, pyrophosphate, and coenzyme A. Common examples of cofactors or metal ions that aid in enzymatic reactions are iron, zinc, cobalt, copper, magnesium, selenium, and manganese. Now, we go to isozymes. What are isozymes? Isoenzymes or isozymes are enzymes that differ in amino acid sequence yet catalyze the same reaction. This difference in sequence can be in one or two or more amino acids. They can be distinguished by one another by biochemical properties such as electrophoretic mobility on a polyacrylamide gel, and also are structurally and immunologically different. We will take the classic example of lactate dehydrogenase, an enzyme that functions in anaerobic or oxygen free glucose metabolism and glucose synthesis. Human beings have two isozymic polypeptide chains for this enzyme. The H isozyme, which is highly expressed in the heart, and the M isozyme, which is found in the skeletal muscle. The amino acid sequences of these two are about 75% identical. The functional enzyme is tetrameric in the sense it has 4 subunits, and many different combinations of these two subunits are possible. The M4 isozyme functions optimally in an anaerobic environment, whereas the H4 enzyme does so in an aerobic environment. This picture denotes the different types of the isozymes of lactate dehydrogenase. H4 is depicted in green, having all 4 subunits of the H type. H3M has 3 H type subunits and 1 M type subunit. M3H has 3 M type subunits and 1 H type subunits. H2M2 has 2 H type and 2 M type subunits, and M4 has all 4 M type subunits. The appearance of some of these isozymes in the blood are indicative of tissue damage and can be used for clinical diagnosis of certain conditions. For instance, An increase in levels of serum H4 relative to H3M is an indication of myocardial infarction, which is nothing but a heart attack. A central approach to studying mechanism of an enzyme catalyzed reaction is to determine the rate of the reaction and how it changes in response to changes in experimental parameters. This discipline is known as enzyme kinetics. It is the oldest approach to understanding the mechanism of an enzyme catalyzed reaction. It is chiefly dependent upon substrate concentration, which is denoted in square brackets. Studying the effects of substrate concentration is difficult because during the course of reaction, it is very quickly converted to product. A simplified approach is to measure the initial velocity or initial rate of enzyme reaction catalyzed as V0, when substrate concentration is far greater than enzyme concentration, which is capital E denoted in square brackets. The effect of substrate concentration on velocity when the enzyme concentration is held constant shows that at relatively very low substrate concentration, the V0 or initial velocity increases very rapidly and linearly with an increase in substrate concentration. At much higher substrate concentrations, as you keep on increasing the concentration of substrate, V0 or initial velocity increases by smaller and smaller amounts in response to increase in substrate concentration. And therein a point is reached where further increases in velocity becomes vanishingly small as substrate concentration increases further, meaning it plateaus. This plateau like region is close to the maximum velocity that an reaction can reach. So, before the substrate is converted to product, it is held in an enzyme substrate complex, the ES, which is the key to understanding this kinetic behavior. The understanding that the ES complex is a necessary step to catalysis was expanded into a general theory of enzyme action by Leonore Michaelis and Maud Menton in the year 1913. These are the two scientists who have first discovered the enzyme catalysis and kinetics. They postulated that the enzyme first combines reversibly, remember reversibly, with the substrate to form a complex in a relatively fast reversible step. So, you can see that enzyme plus substrate gives enzyme substrate complex, which is a very fast reaction, and this is the first half of the complete reaction. The rate constant of the forward reaction is denoted as K1 and the rate constant of the reverse reaction is denoted as K minus 1. In the second step, the complex breaks down into a much slower second step to form the product. ES is converted to E plus P, which is again reversible. The rate constant of the forward reaction here is K2 and the rate constant of the reverse reaction is K minus 2. And after the complex breaks down to yield the product or P, the enzyme is again regenerated and free to catalyze the reaction with another substrate molecule. So, the relationship between the substrate and the concentration and the reaction rate can be expressed quantitatively for measurement, and the curve expressing the relationship between substrate concentration and velocity or rate of the reaction has the same general hyperbolic shape. And algebraically, this can be expressed as the Michaelis-Menten equation named after the scientist who discovered it. V0 is equal to Vmax into substrate concentration divided by Km plus substrate concentration. Now, what is this Km that has suddenly appeared? Km is nothing but the Michaelis constant. All of these can be measured experimentally. The equation has been derived algebraically, and it is the rate equation for one enzyme, one substrate enzyme catalyzed reaction. It is statement of the quantitative relationship between the initial velocity V0, the maximum velocity Vmax, the initial substrate concentration, and all related through the constant Km. This Km has units of concentration. It has been derived from K minus 1 plus K2 by K1. We have already seen what K minus 1, K2 and K1 are. They are rate constants of the enzymatic reaction. So, K minus 1 plus K2 by K1 is nothing but Km. For example, at very low substrate concentration, Km is much more than substrate concentration, and therefore, the term substrate concentration in the denominator becomes insignificant. The reaction then simplifies to V0 is equal to Vmax plus S by Km, and V0 exhibits a linear dependence on substrate concentration, as can be seen in the figure. In the opposite situation, where the substrate concentration is very high and is much more than Km, then Km in the denominator becomes insignificant, and the equation simplifies to V0 is equal to Vmax, because the substrate concentration in the numerator and the denominator get cancelled out. And an important numerical relationship that emerged from this equation is the special case where V0 is exactly 1/2 of Vmax. So, if Vmax by 2 is equal to Vmax into S by Km plus S, on dividing by Vmax, we obtain 1 by Km plus S is equal to S by 2. So, when you solve the equation, it is derived that at exactly half maximal velocity, the substrate concentration that is required at this particular time point is called Km. So, Km can be defined as substrate concentration at half maximal velocity. Enzyme inhibitors are molecular agents that interfere with catalysis, either slowing or halting the reactions. The activity of many enzymes can be inhibited by binding of specific small molecules or ions. This means of inhibiting enzyme activity serves as a major control mechanism or regulatory mechanism in biological systems. So, enzymes catalyze virtually all cellular processes. So, all inhibitors are the most important pharmaceutical agents. For example, aspirin, acetyl salicylate, these are painkillers which inhibit the enzyme that catalyzes the first step that synthesizes prostaglandins. These prostaglandins are involved in many processes which involve pain within the body. The study of enzyme inhibitors provides valuable information about enzyme mechanisms also, and has also helped define a number of metabolic pathways and understand these metabolic pathways. There are two broad classes of inhibitors. One class is the reversible inhibitor, the other class are the irreversible inhibitors, which are generally chemical poisons that kill an individual. Let us look at reversible inhibition first. This can be competitive, non-competitive or mixed. By competitive, what do we mean? Look at this picture, you can see that the substrate and the competitive inhibitor have similar structures in the region that binds to the active site. So, the competitive inhibitor can compete with the substrate and bind to the enzyme's active site. In this condition, if you add more and more substrate, it can displace the competitive inhibitor and the enzyme reaction will proceed to breaking down into product. So, essentially the competitive inhibitor has slowed down the enzyme catalysis. In a situation where inhibitor concentration is much more than substrate, then the reaction will be completely inhibited or completely slowed down till more substrate is added that can replace the inhibitor. Now, you look at non-competitive inhibition. In non-competitive inhibition, what happens is the enzyme binds to the substrate first and forms the enzyme substrate complex, and then the inhibitor binds at a site apart from the active site and therefore slows down the rate of the reaction. Now, we go to the second type of inhibition called the irreversible inhibition. These combine with or destroy a functional group on the enzyme which is essential for its activity or forms a particularly stable covalent association. Formation of a covalent link between an irreversible inhibitor and an enzyme is common. If you look at this structure, the yellow line indicates the active site of the enzyme and the OH group is the functional group projecting out from the amino acid serine. The inhibitor is forming a covalent association with the O of the OH. The OH is released and the fluorine portion of the inhibitor is also released. So, the irreversible inhibitor has completely inactivated the enzyme active site by making the OH unavailable, and therefore, this enzyme is no longer able to catalyze a reaction. Carbon monoxide and many such poisons are examples of irreversible inhibitors which block respiratory reactions and result in death. The last type of inhibition, a completely different type of inhibition, is allosteric inhibition, which is essential to regulatory enzymes. In allosteric inhibition, an enzyme has a secondary active site called the allosteric site, and this allosteric inhibitor or modifier binds to this allosteric site and closes or distorts the active site. So, you understand here that binding of the modifier or allosteric inhibitor to a site apart from the active site changes the conformation of the active site in such a manner that it is no longer specific to the substrate and can no longer bind to the substrate. This prevents the formation of the ES or enzyme substrate complex. In allosteric activation, the activator binds to the allosteric site, allowing the active site to open up and bind the substrate much better than the previous active site. To summarize, I think we all understood from today's class that enzymes are inherent to all life's processes, and without enzymes and the reactions that they catalyze, life itself does not exist. Thank you and hope you enjoyed today's class.